Okay, hi guys. I forgot to record the first half of our meeting or our lecture today. So that's all right. Luckily, we just covered one topic during that time. However, it was a very important topic. So I'm going to go ahead and cover it now. So I apologize. But it'll be fine. Okay, so just to kind of pick up where we left off last Tuesday. 2 days have meaning anymore. Only when we have lecture. So we did go through this question. So just to kind of remind us where we left off on Tuesday. On Tuesday, we did talk about how we can regulate transcription basically by making DNA accessible or not. So we talked about doing this chemically via methylation. And just a reminder that there are two things that we can methylate to regulate transcription. We can methylate histones, or we can methylate individual nucleotides. So with this question, just to kind of recall that lecture, how does DNA methylation, so this doesn't specify necessarily whether we're talking about histones or nucleotides, although my guess is that saying DNA methylation means nucleotide methylation. Regardless, how does methylation affect gene expression? Does it A, enhance transcription? Does it B, reduce transcription? Or C, does it have no effect on transcription at all and instead affect translation? So to answer this question, let's go back to, I'm just gonna turn my sound off for this so you don't have to listen to my emails come in. To answer this question, I do want to revisit two things. So first, revisit this slide where you can see that for if we were to have methylation of histones, what happens is DNA, the more methyl groups so we have on those histones, the more tightly DNA is wrapped around those histones. So what this means is the more tightly wound DNA is, the less accessible it is to RNA polymerase in order for it to bind and transcribe mRNA. So what that means is that more methylation of histones closes DNA, silences it. So if you have it silenced, that's less transcription. Same thing goes here with epigenetic changes. So you can see here that if you have more nucleotides with methyl groups attached to them, it turns the gene off. And the reason this is because whereas the normal nucleotide, like I use the example cytosine because that's the nucleotide that's kind of like most methylated, at least if I'm remembering that correctly. But cytosine in its normal state is recognized by RNA polymerase. But if you methylate that cytosine, that changes it enough to where it's not recognized by RNA polymerase anymore. So that means that RNA polymerase can't see a cytosine or a C, and therefore create its complementary base, a G, in an mRNA strand. So if that means that the more methyl groups you have on individual nucleotides, that's also going to halt transcription because RNA polymerase isn't going to be able to recognize those nucleotides for transcription. So getting back to our question, then how does DNA methylation affect gene expression? Either by tightening up how DNA is packaged or by methylating individual nucleotides, it would reduce transcription. Right, so let's move on, shall we? All right, so we talked about, we started talking about gene regulation on Tuesday, and I just want to mention that for the rest of this lecture, what we're kind of going to do is kind of go through this sequentially And what we're going to start with is right here, number 1. And again, I want to mention that I don't really necessarily like the term most important level of gene expression because it's a little misleading. All gene regulation is important. But as you'll find out for what we're going to talk about first, which is transcriptional control via transcription factors, is arguably the most important. Not necessarily because it's like the best level of control, though it's very, very good, but it is the most ubiquitous. So we're going to talk about a lot of places that gene expression can be regulated, but every single one of your genes is regulated at that first step. So not all of your genes will be regulated at steps 2 through 6, several of them are, but all of your genes are regulated at this level. And they are all regulated at that level by what are called transcription factors. So what are transcription factors? So first off, in order to understand what a transcription factor is, we do have to kind of look a little more closely at the components of the promoter site, which we talked about earlier, so a while ago now. And if you remember, the promoter of a gene is the start site for transcription. So the promoter, remember, a gene is basically a sequence of nucleotides from the start promoter to the end, which is the terminator. And the big thing that we said happens at the promoter, the reason it's the start site, is because that's where RNA polymerase binds. So if RNA polymerase binds the promoter, that's where we start transcription. That's not necessarily the whole story. So there's a lot more to basically starting transcription than just having RNA polymerase bind. So we also have the promoter. So you can see up here, this top picture, what you're looking at is in pink, kind of here, it says protein encoding gene. So that would be the sequence of nucleotides that will code for mRNA. And then you see in this sort of like teal color, that is your promoter. But you see that technically in this slide, it's called a core promoter. And that's because aside from what we talked about, which is the transcriptional start site, which is where RNA polymerase binds, there are some other components. And a really important part of that is what is called the Tata Box. So the Tata Box is called the Tata Box because it's created mostly out of nucleotides T and A. And you're always going to see that sequence within the Tata Box, TA. So you'll see those 4 nucleotides in that sequence. And then after that, it's usually going to be some other T's and A's. So they'll all be Tata. Some will be Tata, like this one, and others may be Ta. But regardless, you'll always have that 4 nucleotide sequence, which is going to be Tata. And that is upstream, or what that means is just it comes before the transcriptional start site and the actual gene. So the combination of the transcriptional start site, which is where RNA polymerase binds, and the Tata box is what we call the core promoter. Okay, so what happens at the core promoter? Well, we already know that at the transcriptional start site, that's where our RNA polymerase binds, shown in purple. But you can see that at the Tata box, you also have these kind of like yellowy proteins, and those are called transcription factors. So there's a lot of transcription factors. They can basically function to turn genes on or turn genes off. They're usually named based on what they do. So for example, activators activate transcription, they turn transcription on. Inhibitors inhibit transcription, which means they turn transcription off. Why is it doing that? Whatever. But they're named based on how they affect transcription. So the example I was giving this morning when I went through this lecture is the purpose of transcription factors are kind of to basically like, we know that there are genes that need to be transcribed, but transcription factors basically give us the go-ahead for transcription. So we will look at this, but we kind of talked about before with transcription, what happens is you'll just have RNA polymerase bind to the promoter and then you get transcription. And it's actually quite a bit more complex than that, because as you can see in this picture, RNA polymerase is not the only protein that binds to the promoter. You also have those transcription factors. So with this, if you think about the fact that, like, if you use the analogy, right, RNA polymerase reading DNA into mRNA, because RNA polymerase is moving down the DNA strand, you can think of that as like driving your car, right? So when you drive your car, may seem pretty straightforward, but there's actually a lot of things that need to be in place in order to drive your car. First, you need to have a place to go, right? If you have to go to work, you have to be there on time, right? So there's a certain time, right? You need the go-ahead to go there. Second, you need to be able to start your car, turn it on, and you need to be able to push the gas pedals to say go, or brake, right? Gas or brake. So that's kind of what we're talking about with transcription factors, and those are kind of the levels of control we have here. So what we have with this is basically you can think of RNA polymerase as your car itself going, and you can think of these transcription factors as basically like the keys to your car and turning your car, like having the engine on, okay? So if RNA polymerase is bound to a promoter, all you have is a car. The first thing you have to do is turn the car on, turn the engine on. That's what happens when those transcription factors bind to the Tata Box, right? So you're sitting in the car, you turn the car on. Now you're ready to go. And the next thing you have to do though is press either the gas or the brake. So that's a little bit more complex. So if you have your car on, or you have RNA polymerase on, that is what is called the pre-initiation complex. Basically, RNA polymerase is ready to go. So the second thing that has to happen again is you have to basically give it the signal to go or stop, versus press the gas or press the brake. And that happens via what are called enhancers. So enhancers are not directly associated with this core promoter. They are part of the overall promoter. They're actually a little bit upstream or outside of that core promoter, just as another level of regulation. Basically, like distance from the core promoter means that if you have that entire pre-initiation complex bound to the core promoter, pressing stop or pressing go or pressing the gas or pressing the brake is still kind of just like out of the control of that initiation complex. It's like having only your feet next to the gas or brake, as opposed to like the other people in your car having their feet in the same area. So it's basically like regulation and safety by control. All right, so we have in the pre-initiation complex, you have your RNA polymerase bound, and you have transcription factors bound. So your car is on. So how do you give it the signal for gas or brake? And that's via those enhancers. So that's what you're looking at in this slide. So looking at a different view of this. At the top of this slide, what you're looking at is your DNA. And here in green, you have your core promoter. So remember that is your RNA start site as well as your Tata Box. And then after that, you have your gene, which is all the nucleotides that are going to be transcribed into mRNA. So remember, at the promoter, which you can see down here, at the promoter, you have a couple of things bind. You have your RNA polymerase bind, which is shown in gray. And then you also have those other transcription factors. This is called like DNA binding protein, which we'll talk about in a minute. And then you have other proteins. These are transcription factors. Those also bind to the Tata Box as well as RNA polymerase. Okay, So again, upstream of the core promoter, you have regions of DNA called enhancers. And what binds to those enhancers are activator proteins. And what happens when those activator proteins bind to these enhancers is this region of DNA actually bends over and allows those activator proteins to associate with the pre-initiation complex. And that is when you basically press the gas. Okay, so you can see that kind of here on the bottom of your slide, where you have your gene, you need to transcribe into mRNA. When you step in your car, so again, your car is the RNA polymerase, and when you start your car, that's the assembly of all those purple transcription factors. And then what has to happen in order for your car to go is you have to have those activators bound to this as well, which, as you can see, actually requires the DNA to kind of bend and have those activators associate with that entire complex. So we actually did watch this process previously when we watched that awesome video of hemoglobin being made. And so I like to look at that video again, and now that we know all of the components that are required for transcription, kind of look more closely at what is happening here. So you remember this video, we're just going to look at it again, but this shows not only RNA polymerase binding, but it also shows the pre-initiation complex, all of the transcription factors, and the initiator, and it shows the bending of DNA. So let's take a look. Whoops, I need to have my volume on. Here we go.
What you are about to see is DNA's most extraordinary secret.
Not a secret
the
anymore.
The innermost workings of how a simple code is turned into flesh and blood.
So first off, you should note that what is attached to the DNA, so the DNA is red, what's attached to the DNA right now is not RNA polymerase. That is a transcription factor. So the only thing bound to the core promoter, so this is going to be bound to the Tata Box, is a transcription factor right now.
Is what Francis Crick called the central dogma of modern biology. How DNA makes protein.
You'll see some stuff come in.
It starts with a bundle of factors assembling at the start of a gene.
The promoter.
It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein.
Okay, so that blue, what just flew in and bound, is your RNA polymerase. So here we have a transcription factor bound, and we have RNA polymerase bound, and what you'll see is several other transcription factors also come in.
The gene is the length of DNA stretching away to the left.
Okay.
Everything's ready to roll.
That's your pre-initiation complex.
2, 1.
Okay, so what you're seeing is you're seeing that DNA bend. Right here would be the enhancer region, and one of those activator proteins would be bound to the enhancer region. So in order for the RNA polymerase to be released from this pre-initiation complex and to go to transcribe, what we have to have is we have to have that activator bind to this pre-initiation complex. So again, here you're sitting in your car is on. All you have to do is press the gas, and that's what this activator does. So you can see it, here we go. Right, so notice that was very important. The big thing there is that, a little bit back, that's where I want. You'll see that once the activator bound, it released RNA polymerase. RNA polymerase is the only thing that moves along DNA to transcribe RNA. All of those transcription factors and everything are left at the core promoter. So RNA polymerase is the only thing that is transcribing. But all of this stuff, the transcription factors and the activator, all of that has to be present to basically just tell it to go. You have to have the key to your car has to be on, you have to press the gas. But once you do all that stuff, you're going. And you can watch the rest of that video too. It is linked on this slide as well. I encourage you to watch it repeatedly just because it's cool. All right, so I also have another video. This is another really good video kind of showing, you can see in yellow, the enhancers, you can see the promoter, the core promoter, the Tata Box, and you can see the coding region. So this video also shows that process. I encourage you to watch it. It's in the PowerPoint that is on Canvas. Just for the sake of time, I didn't go through it. But let's go ahead and go through it because I have time. I'm recording this. Okay, let's go. Right.
For RNA polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short TATA sequence in the promoter.
Those are transcription factors.
Other basal factor proteins then bind, eventually forming a full transcription factor complex able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors, called coactivators, link the basal factors with a third set of transcription factor proteins called activators. Activators are regulatory proteins that bind to sequences on DNA called enhancers. Enhancers are located at sites that are distant from the promoter. The interaction of activator proteins with transcription factor subunits increases the rate of transcription. Many enhancers scattered around the chromosome can bind different activators, which provide a variety of responses to various signals. When a second kind of regulatory protein called a repressor binds to a silencer sequence located adjacent to or overlapping an enhancer sequence, the corresponding activator is no longer able to bind to the DNA.
This is a pretty good video. Showed the repressor protein, which we didn't actually talk about. Sometimes those are also called inhibitors. But again, you can see that if the activator protein can't bind to the pre-initiation complex, then RNA polymerase is not going to go. So this went into a little bit more depth than we did, but I think it was still pretty good. So again, this is really important. And the reason I think this is so important to emphasize is because if we look at this slide, there are lots and lots and lots and lots of places where the process of turning gene into protein can be regulated. Really, like, you know, we're going to talk about all of these, but not all of your genes do RNA processing control or transport or degrade. All of these, a lot of your genes do, but not all of them do. But all of your genes are regulated by transcription factors. So that's why we say it's like the most important. I don't know if it's like the most important, but it certainly is the most ubiquitous. All of your genes are regulated in this way. So very important for that reason. So you do need to be sure that you understand transcription factors, the purpose of the Tata Box, which is where transcription factors bind, that RNA polymerase binds to the core promoter, that those enhancer regions are where your activators bind, and that, yeah, kind of everything we talked about. So that's really important stuff. Here's a good question to kind of sum this. What would be the action of a transcription factor that functions as an activator? So remember that what we're doing via transcription, we're turning DNA into RNA, and activators are proteins that basically make this process go. So obviously A would be incorrect because it would not inhibit, right? That's the opposite of activate, so that's wrong. This is wrong because it has nothing to do with ribosomes. This is transcription, not translation. And we didn't even talk anything about operons, so you need to know that. So the logical answer is B, and the activator of transcription. If it's activating transcription, the purpose of that is to take DNA and turn it into RNA. So what you're going to do is increase the amount of RNA created. Awesome. Okay. So if you have any questions, please be sure to let me know. And yeah, good job.